Technology conference. Do you hear me?
Yes.
Yes.
Okay.
Yes.
I would like to thank the organizers of the World Biological Science and Technology Conference 2021 for inviting me to give a keynote speech at the plenary session. The title of my speech is Remote Bioholography, Restoring the Wave Optics of Chromosomes Damaged by the SARS-CoV-2 Caused Autoimmunity. I'm the author of the fundamental theory of wave optics in chromosomes and the founder and president of the Wave Genome LLC biotechnological company founded in 2010. The title of my speech is The Remote Bioholography for Restoring the Wave Optics in Chromosomes Damaged by the SARS-CoV-2 Caused Autoimmunity. All proteins in SARS-CoV-2 are homologous to human proteins, except for one. Homology means that nucleotides fit into each other as a key and a keyhole by the principle of complementarity. That is why COVID-19 causes the destructive RNA interference with the resultant autoimmunity and sterilization. The effect of the destructive RNA interference is the cause for the enhanced immune response, pathogenic priming, ADE, antibody-dependent enhancements, cytokine storm, and finally AIDS. Andrew Fire and Craig Mello received Nobel Prize in 2006 for the discovery of the effect of the destructive RNA interference. They took RNA from C. elegans worm and gave it back to the worm, and the worm died. Thus, in order to treat COVID-19, we need to eliminate the effect of the destructive RNA interference. All treatments, which like mRNA vaccines, introduced even more homologous RNA into a human cell, will inflict more damage due to the effect of the destructive RNA interference. Even more, COVID-19 mRNA vaccines are proven to cause the prion disease when the proteins lose their crystalline structure, acquiring a sponge-like shape as the result of the structural damage in chromosomes. It is proven that SARS-CoV-2 is capable of inserting its genetic material into human DNA, so that SARS-CoV-2 is a chimeric virus with the gain of function, a retrovirus under the disguise of a coronavirus. So first of all, translation of proteins is destroyed by the destructive interaction between the homologous RNA in vaccine and the RNA in a human cell. Secondly, due to their homologous nature, vaccines' nucleotides get inserted into human DNA so that a human becomes a walking factory producing a virus. This actually was the idea from the start, and it got called Safe Gene or Gene Drive Biotech by DARPA to make people produce their own vaccines, antibodies, by making people produce the virus on the constant basis. But because virus nucleotides got inserted into human DNA, it turns out that man's immune system attacks itself, causing AIDS, the autoimmune disease. Thus, we need to characterize the gain of function and dual-use research of concern SARS-CoV-2 as the airborne AIDS and mRNA vaccines as the prion priming. The COVID-19 global event gives us an opportunity to rethink the rationale for the gene editing via the homologous nucleotides and the reverse transcriptase, transcriptase, dsRNA, treatments via client's own stem cells, and as a whole, the rationale for the synthetic computational biology which violates the structural coherence of chromosomes via adding and subtracting. We should realize once and for all that the genetically modified humans will be as sterile as the genetically modified plants. Even more, if we are not going to deal with the consequences of the gain-of-function SARS-CoV-2 and COVID-19 mRNA vaccines, the humankind will experience the genetic collapse and the extinction of human species. At the moment, the world is on the verge of the global health system collapse due to the impending global prion disease after the 8-year incubation period. Prion disease is the most severe case of encephalopathy. The mass prion disease will cause the total failure of political and social system. With Genome LLC found a technological solution, the Remote Holographic Drug Delivery, for reversal of the structural damage in chromosomes and so the reversal of the structural damage in proteins. Humans are water for 85%, but the universe is energy for 93% and particles for only 7%. So humans are the liquid crystal media for 7% and the wave crystal media for 93%. In order to restore the structural coherence in the liquid crystal media, we need first to restore the structural coherence in the wave crystal media. The notion of the wave crystal media, wave crystal, a crystal consisting of waves, was created by me in 1985, long before anybody in the field of bioholography, in my fundamental theory of wave optics and chromosomes. Wave crystal is a refractive lens built up by the coherent impulse wave fronts of the scalar nature, with the trigger signal having its wavelength equal to the size of the resonator. Between cell division cycles, chromatin exists in non-coherent state, but during cell division, chromatin gets literally crystallized, forming two chromatids, which form a chromosome. The structural coherence of the chromosomes determines its functionality. The chromosome consists of two chromatids combined together in the X form. The two chromatids intersect in the center where they exchange their genetic material. The same gene is expressed in the functional individuals by a metacentric chromosome and in dysfunctional individuals by acrocentric chromosome. Here we see on the left the acrocentric chromosome, which is literally half of the metacentric chromosome, and on the right, the metacentric chromosome. Metacentric chromosome are the egg-shaped chromosomes with two arms that are equal in length. Acrocentric chromosome are chromosomes with two arms that are not equal in length, meaning they exhibit gross asymmetry. Metacentric chromosome is analogous to a well-centered and focused eye crystal, while acrocentric chromosome is analogous to a myopic or farsighted eye. Ability of chromatids to exchange information and to regenerate and replicate in general depends upon the specific geometry of wave optics in the chromosome. If this specific geometry of wave optics is violated, the chromosome loses its ability to regenerate and replicate. The same gene in apes is expressed by the acrocentric chromosome and in humans by the metacentric chromosome. The same is with gene in horses. The same gene is expressed by the acrocentric chromosome in the wild horses, and the same gene is expressed by the metacentric chromosome in the domesticated horses, which are superior to the wild horses in physical and intellectual abilities. Children with Down syndrome differ from normal children by the presence of extra acrocentric chromosome. Only because female gender chromosome is metacentric, a woman is capable of replicating, giving birth, and only because male gender chromosome is acrocentric, a man is incapable of replicating, giving birth. Here we see on the left the male chromosome, which is literally the half of the female gender chromosome, and on the right the female gender chromosome, which is properly regenerative and functional, and male chromosome is degenerative and dysfunctional. Clearly, the acrocentric chromosome is the result of the destruction of the metacentric chromosome, literally splitting into halves. Evidently, Y, male gender chromosome, which is acrocentric, is the result of degeneration of X, female gender chromosome, which is properly metacentric. Structural coherence of the chromosome is its physical shape for 7%, while for 93%, it is the proper wave optics of the scalar wave diffraction grating with properly centered and focused refraction towards the zero center focus of the wave crystal. Zero center is the term of the quantum physics. Chromosome is a wave crystal with certain wave optics and changes its bioinformation due to the change in its refraction mode towards one and only zero focus or zero center, the same for chromosome and galaxy. Chromosome is a lens with the refraction code unique for a given individual. So we can understand human genome only in terms of wave phenomena as it truly exists, as the stages of crystallization of the liquid crystal media correspondent to the stages of crystallization of the wave crystal media. The crystallization of the wave crystal media is the emergence of its bioholographic quality. The first stage is the destructive and constructive wave interference. Wave interference in the living cell results in polarization due to the change of spin in the energy torsion processes. Human RNAs are the one-strand spiral antennae susceptible to homologous viruses due to the spatial translational symmetry principle of physics. In electron microscopy, RNA appears to be the simultaneous torsion firings across the cell. It was reported, though not explained, by Jennifer Doudna that the application of CRISPR-Cas9 technology to the living cell had the result of extinguishing the wave activity of RNA. 93% of functionality of human genome. The effect of the destructive RNA interference is mainly the effect of the destructive wave interference. The second stage is the reflection of waves back upon themselves due to the polarization in resonators correspondent in their size to the wavelength to form a scalar wave. Scalar wave annuls the external linear signals when the peak of the forward-going wave is nullified by the trough of the same wave when this wave is reflected back upon itself. Here we see the scalar wave, which emerges when the signal is the impulse coherent signal. Coherent means it preserves its frequency permanent. The third stage is the formation of the scalar wave diffraction grating and its scaling when every segment of the scalar wave diffraction grating itself becomes a scalar wave, like this. This third stage is the emergence of refraction, so that the linear external signal changes its nature and its frequency going through every segment of the scalar wave diffraction grating. The signal changes its frequency. This means it's getting refracted, and it changes its nature, thus becoming a holographic signal. Holographic signal contains inside itself the infinite number of waves, frequencies, particles, and fields due to the non-locality of the scalar wave diffraction grating. This is all quantum physics. Scalar wave diffraction grating gets scaled into the macro and micro dimension. That is why it is used to transfer bioinformation instantaneously and remotely at close and long range, including in the human body. Quantum nonlocality is organized in accordance with the holographic principle. The holographic principle states that universe is entirely in its every matrix point. This means quantum nonlocality. Implication of the holographic principle formulated by me in 2012 states that if the universe is entirely in its every matrix point, then every matrix point is not simply different from any other matrix point, but is unique. Thus, every matrix point or wave matrix or biohologram is both non-local and unique. If you have two copies of the same wave matrix, we can transmit information remotely and instantaneously between these two copies. In quantum nonlocality, they constitute one and the same entity, the systematic whole. All processes of refraction in the wave crystal media occur toward one and only 0 center of quantum nonlocality, one and the same for galaxy and chromosome. But each wave matrix has its own unique refraction code. Thus, gene therapy is possible only through correction of the wave optics in chromosomes, while restoring the refraction code unique for every human. Tokens with the same refraction code constitute one and the same non-local and unique systematic whole in the quantum nonlocality. Instead of the vague term quantum entanglement, I offer the term refraction entanglement as the basis for the instantaneous and remote transmission of bioinformation between the tokens having one and the same refraction code of their wave optics. So DNA is a tetrapolar scalar wave which annuls the external linear signals when the peak of the forward-going wave is annulled by the trough of the same wave when this wave is reflected back upon itself. DNA scalar wave is continually annulling the simultaneous RNA wave torsions across the cell. The quality of transcription and translation depends on the coherence of the DNA scalar wave. I repeat, the resultant scalar wave diffraction grating has certain refraction code, unique and non-local for every human. DNA communication occurs not through chemical and electrochemical exchange between living cells, but via the quantum nonlocality, remotely and instantaneously. Wave Genome LLC, funded by me and founded, was the first company in the world to develop bioholography as the remote wave optics based upon refraction codes in the scaled scalar wave diffraction grating. Refraction codes are used as the wave modulations of the scalar nature as client's unique and non-local biohologram, immune system modulation biohologram, stem cell modulation biohologram, etc. Wave Genome LLC has pioneered bioholography, producing bioholograms for the remote management of biosystems. Wave Genome was the first company in the world to code electrodes with clients' unique bioholograms. Wave Genome was the first company in the world to produce bioholographic electronic devices, digital files, and software for the holographic drug delivery for rejuvenation only. I co-founded the Quantum Biointernet for the remote rejuvenation via distant laser signal of a special nature, commercially offered for the first time in the history of humankind in May 2013. I co-founded the Matrix City Consortium with the Institute for National Security in Moscow for building self-sufficient human settlements based upon not graphene oxide, but based upon quantum leap bioholography, and having the remote management of biosystems, climate, and geophysical processes for the first time in the history of humankind, founded 2012. Presented in the same year to Dr. Putin's President Putin's inner circle, and presented as the honorary lecture at the Harriman Institute of the Columbia University in September 2012. Reproduction and generation success depends on the properly focused wave optics of chromosomes for receiving and transmitting the non-local unique bioinformation from quantum biononlocality of the certain biohologram. So my company offers electrode-based chips structured on nano-level via laser by client's digitized hologram modulated by various modulators, including stem cells, and produced in cooperation between Wave Genome LLC and Bauman Technical University in Moscow, the largest and most important science university in Russia, with scientists who have produced mathematical models for trajectories of the latest Russian middle range, translation from Russian, middle range missile caliber. Digital bioholograms of healthy physiological systems of stem cells. Wave Genome has produced the holographic stem cell also in collaboration with Bauman Technical University. The stem cell was extracted from the epithelial cell and in the electron microscope, and then recorded as a biohologram in the special non-optical laser at the Bauman Technical University, and then produced the digitized holographic stem cell and used as a modulator for the large size generator of the scalar wave diffraction grating at the Bauman State Technical University. So we have the whole pharmacy of holographic drugs and the protocols of their remote at instantaneous delivery called HoloNet, holographic by internet, founded by my company in 2013 for the remote treatment. So Wave Genome LLC was the first company in the world to record and transmit the digital biohologram of various kinds of stem cells, including the stem cells extracted from epithelial cells and embryonic stem cells, in collaboration with the Bauman State Technical University for the holographic pharmacy and for the remote stem cell therapy. And we were the first company to commercially offer the remote stem cell therapy through the quantum nonlocality via non-optical lasers capable of creating the holographic signal. Technology of the laser polarized bioholography for the HoloNet, holographic internet, consists in the know-how for creating the holographic signal, which is in principle non-local in time and space, and second, in modulating this holographic signal via various modulators, including the digital biohologram of the stem cell. We can treat the cell degeneration leading to AIDS and prion disease as the result of COVID-19 global event by applying the holographic signal. I have introduced the new method for recording and remote transmitting of bioholograms via laser spectroscopy and coding electrodes on nano-level via laser. And it has certain stages precisely of creating the scalar wave based upon binary mechanism of our biological processes. So we have 2 eyes, 2 ears, 2 lungs, precisely because our brain needs to produce the scalar wave. So I have produced lasers and I have produced large size generators precisely built upon these processes. So this is not just scalar wave. It is this technology with all the details of how to produce the proper dense scalar wave diffraction grating. The technology creates lenses assisting our chromosomes in correcting their refraction for focusing genetic bioholographic information both locally and remotely. The same way we use lenses to correct eyesight, we can use artificial lenses to correct geometrical optics of chromosome. And I have conducted clinical trials on the basis of the Bauman State Technical University in Moscow for holographic aspirin in 2017, successful clinical trials. And trials used the electret-based generators or applicators and the large size generator. And that's it. Thank you very much. And I hope that we will cope with the global event COVID-19, and we will find solution to the severity of autoimmune diseases which are coming after the mass mRNA vaccination, as was proven by the scientific research. Thank you very much.
Thank you very much for Dr. Caesar for your wonderful talk, brilliant like information you shared with us. For me, it's a really a brand, like a very like new things. So let's welcome for our Q&A. So if any audience would have a question for Dr. Caesar. Yeah, I would ask around about our audience for any questions. Yeah, are you able to hear me?
I hear you.
Oh yeah, sure. So let's open it for the audience. I think the audience is quiet. Yeah, of course, your research is really brilliant because it's you really touch the one touch the area not many common people understand. So
well, I can if it is a question of this kind, right? I can say that our ancestors used scalar waves for millennia. This is how they coped with viruses and bacteria. And all spiral-shaped bacteria and spirochete bacteria and all spiral-shaped viruses, they cause encephalitis, they cause encephalopathy. Because they get these spiral shapes, they get inserted into our RNA via this spatial symmetry, translational spatial symmetry principle. And our ancestors used electrodes on the daily basis. They used oils, solid form electrodes as resins, and aerosol form of electrodes. The entire Asia, you know, we have here speakers from Japan, from China. The entire Asia survives on liquid electrodes. We have in Russia the notion to anoint for the kingdom, or the anointed one. The king was anointed with the essential oil on his head, because the oil, essential oil, is produced from the resin, and resin is the electrode. What is electrode? Electrode is the artificial or natural crystal which precisely creates the scalar wave. So it does not let the signal through. It gets the signal refracted toward its center. And via creating the scalar wave, it annuls the waveforms of bacteria and viruses. Our famous surgeon Pirogov in the middle of 19th century during the Crimean War, when Russians had protected Crimea from the invasion by British and French, our famous surgeon Pirogov said that one can operate in a 5-meter radius near the Crimean juniper, because juniper produces the resin, which is the electrode. And juniper oil is famous for protection, for giving protection against viral and bacterial infections. And in the Black Plague period in the 15th century, only those people who used frankincense oil were capable of survival. So it is a millennial-long technology. Also, our ancestors used the solid form of electrodes. Our ancestors were wearing the egg-shaped pendants, and inside this pendants was the frankincense as resin in solid form. Also, our ancestors used the aerosol form of electrodes by smoking, burning the incense.
Okay, so, Dr. Caesar, in view of the time, what's the commercial status of your technology? Could you share a little bit? any commercial deployment of your technology right now?
Well, my company is 10 years old. It is a very successful company, making approximately $500,000 a year. So it's a very small company, compact company, and we serve the elite segment of the market, but preparing to bring this technology to the mass market. Because right now right now, so that you understand, we are approaching the technological leap, which will be concerned with the control of the genome via precisely wave technologies, and not just by digital homeopathy and different kind of frequencies. No, precisely by the quantum computers, control of wave interference, also psychotronic weapons, they are based upon the interference of the low and high frequency.
Dr. Caesar, yeah. So if we have other questions, we will send it to you directly. In your time, let's keep on track. So let's move to the next presentation. So the next presentation would be mine. So Nancy, I would like to share the screen. Could you allow me? Thank you, Dr. Caesar.
You're very welcome.